So in lab this week, you are going to be conducting a DNA profiling experiment in which you have to solve a crime. You're going to be given DNA from the crime scene, DNA from one prime suspect, and DNA from a second prime suspect. And what you will have to do is use a gel electrophoresis apparatus to compare the DNA and figure out who the criminal really is. So in this video, I'm going to do a run-through of what the lab will look like so that when you come into lab, you will have a very good idea of how to conduct the procedure independently. So as you're watching this video, please have the lab protocol handout handy because you might want to take notes on that. And feel free to stop at any point if the information becomes a little overwhelming. So let's start with the materials that you're going to be using. First of all, this purple box is your light source, or your power source, and you can adjust the voltage. This is where the electricity for your gel electrophoresis will come from. This is the gel apparatus, or the gel box, where you will put the gel, and it has a lid along with electrodes to connect to the power source. This is your gel tray, and there's a ready-made gel inside. Your DNA samples will be found in microtubes like these. You will be transferring the DNA using a fixed volume pipette, and what that means is that this pipette is automatically set to transfer 40 microliters of DNA. So you don't have to worry about measuring, it'll take exactly what you need. These are the tips for the pipette. This is the stain that you will use to visualize the DNA. This is buffer that we'll come back to later. You're going to put that in the gel box. You will also need water and some trays for destaining. Alright, so let's get started with the procedure. The first thing you're going to have to do, actually, is remove the gel from the gel tray. I will have made the gels for you, but when you get your gel, you're going to have to gently remove the rubber dams at each end so that you don't break the gel. And then you need to remove this gel comb. The comb is used to make the wells at one end of the gel. So to do that, you just want to lift straight up, like that, without breaking the gel. So now that you got this done, you're actually going to want to slide the gel into here. So once you place the gel in, there's really only one way to orient it. You'll see that there's a tab, it slides right in. So now that you've got your gel in the gel box, you're going to add buffer. And you're going to pour about 300 milliliters of buffer. It doesn't have to be exact. The point is that you want to completely submerge the gel. It's also a good idea to pour at one end or the other rather than directly on the gel because you don't want to damage or break the gel. Alright, so I can see from this that my gel is nicely submerged, about 300 milliliters of the buffer used. Alright, so I've got my gel in the tray, in the box, covered in buffer. I am now ready to load the DNA, to transfer the DNA from the tubes into the wells. And this is actually the hardest part, and you'll have the opportunity in lab to practice before you run the real thing. So you'll take the DNA that's to be loaded into well 1, take off the cap, take your pipette, and get a fresh pipette tip. You just press down pretty firmly and it'll stay on. Then to extract the DNA from the tube, you simply push down on the pipette, insert, release, and you'll have exactly 40 microliters in the pipette. So once again, push down, put it in, release, and then take it out. So now here's the tricky part. You need to load these 40 microliters of DNA into the correct well. And the hard part is that you need to put the tip in far enough so that it's actually in the well. But if you go too far, you can puncture the gel and then your DNA won't run through the gel. So this will take a little bit of practice. I find it's helpful to put my elbow on the table so that I have a firm look. Okay, and that one's loaded. Now what you would do is discard this tip because it's contaminated with that DNA source, and you'd get a new tip. And you'd move on to DNA sample 2. And you'll keep doing that until you've loaded all of the DNA samples. You're actually going to have 2 samples from the crime scene, 2 samples from suspect 1, and 2 samples from suspect 2. So once all of those are loaded, you're going to be ready to run your gel. So what you will do is take the gel box lid and place it on so that the black and the red electrodes match. So I'm going to put the black to the black, the red to the red, and then I simply hook it up to my power source. Again, red to red, black to black. Once this is tightly on, this is inserted, I can now plug in my power source. It's important to leave it unplugged just for safety. Alright, so now you're ready to run the gel. As I mentioned before, there's two voltages. You're going to run your gel at 150 volts. So you'll click 150, and it should start running. You'll know that the gel is working because you'll see tiny little bubbles due to the heat from the electrical current. You're going to let the gel run for 15 to 20 minutes. You can monitor it closely and you'll be able to track the dye as the gel moves through. So let's say the 15 to 20 minutes is up. Your DNA has run through the gel. The smallest fragments have gone the furthest, the shortest fragments are stuck near the wells. You're going to now turn off your gel. You're going to disconnect the power, and then you're going to remove the lid from your gel box. And it's a little bit resistant. There we go. Okay. So you've now sorted the DNA based on size. The problem is you're not going to be able to see the DNA very well. You need to stain it. So what we're going to do is we're going to remove the DNA, we're going to remove the gel entirely from the box. And you're going to transfer it to one of your trays. And there's a spatula to help you do this. You may find that your fingers wearing gloves works just fine. So now you're going to take your stain. And at this point you would be wearing gloves because this is called a stain for a good reason. And you're going to pour about 75 to 100 milliliters of stain in here, enough to completely submerge the gel. What the heck, we'll pour it all. Alright, so now you're going to let it sit here for 5 minutes. And during that 5 minutes, the stain is going to bind specifically to the DNA molecules to highlight them for visualization in a few minutes. So let's fast forward a little bit, and let's say that the staining is complete. Now what you're going to do is you are going to transfer the gel to another box. And in this box, you would have water. And you're going to basically de-stain the gel for about 30 minutes. At this point you would add a few hundred milliliters of water and you would let it sit here for 30 minutes, agitating it every few minutes. After the first 15 minutes you're actually going to empty out the water and replace it with fresh water. That'll facilitate the staining process. So another 15 minutes, gently agitating it. It should be properly de-stained. At this point, you can discard the rest of the water, and you'll be ready to take your gel and put it on this light box. And this light box, once it's turned on, will illuminate the DNA bands. And at this point, you'll be able to compare the DNA bands from the crime scene to suspect 1 and suspect 2, and you should be able to come to some interesting conclusions about who actually did the crime. So to be prepared for your lab, what you need to do is read and annotate the handout carefully, watch this video, which clearly you've done because you're listening to these instructions, and complete the online poll. You don't have to worry about prepping your lab notebook because there will be plenty of time during the lab while the gel is running and while the gel is de-staining to do your lab background, your problem, your materials, and your results. All right, so a couple more days and you will get to solve the crime. Thank you.